Yeah. Good morning, everyone. Welcome to our today's seminar. Today, our guest is Dr. Houston Wilson from University of California, Riverside, where he's working as an associate cooperative extension specialist. Houston got his bachelor, PhD, and postdoc, everything from UC Berkeley, and joined as a faculty at UC Riverside in 2017. And since his focus has been in extension and research of IPM in of orchard and vineyard crops, and recently he has been also focusing on hemp pest management, and we are really glad that he is here, and Dr. Amanda Hodges Lab are very honored to host him for today's seminar. And yeah,
Great. Hey, thank you so much for the introduction. Took care of first couple of these slides. Oh, sorry. All right ahead. Yeah, so good morning and thank you for the invitation to come share a little bit about my work in California here. Always really looked up to the University of Florida Department of Entomology. It's a very well recognized and established program. So it's a big honor to be here today. So as mentioned, I'm Houston Wilson. I'm a faculty, cooperative extension faculty at UC Riverside. My lab is actually based about 5 hours north of the Riverside campus at the Kearney Agricultural Research Extension Center. This houses extension faculty from both the Davis, Berkeley, as well as Riverside campus. So we've got pathologists and irrigation people, soil people, entomologists. So right there in the heart of the San Joaquin Valley, major agricultural region in California. Our research spans kind of the landscape to the molecular. We do a number of different things. This isn't in any sort of prioritized order, but really everything focused around the development of integrated pest management practices for deciduous perennial crops. So primarily almonds and pistachios, although we do work with walnuts, figs, and grapes. That's where I got my start as a graduate student. So that was starting back in the summer of 2017. And then in addition to that, I was appointed as the founding director of the UC Organic Agriculture Institute in 2020. And this was less about me as an entomologist and more about facilitating research, extension, and education in support of organic agriculture in California, which is the biggest organic ag state in the nation. So for whatever reason, I guess probably a little bit younger and dumber, I took on a second full-time job in addition to being a research entomologist. But we were able to launch this institute. It's a brand new statewide program, as I mentioned, and I actually was able to just step down in order for us to hire a full-time salaried director to oversee this program. And this was an effort that was catalyzed by Cliff Barr, who went around the country setting up 5 endowments to support organic agriculture. So there I am with a few of those chairs. We've got one at University of Wisconsin, Madison, one at Washington State, Tuskegee University, in addition to University of California. So back to tree nuts, though, in California. It's the main thrust of my work presentation today. So these are major crops in California. This map is showing you the little black dots. That's where you have either walnut, pistachio, or almond planted. We've got over 2 million acres in the state. It's 23% of the total cultivated area, much of which is exported. So these are the major commodities in our state. There's multiple insect pests that affect these crops, some of which, like navel orange worm and leaf-footed bugs, stink bugs, they move between these different tree nuts. Others, like Gill's mealybug, that's really specific to pistachio, walnut huskfly, imagine they're specific to walnuts. And then, of course, we have invasive insects. So the Carpophilus beetle, we discovered this thing infesting almonds and pistachios in California back in 2020. So a wide range of stuff to deal with here. What I'm going to focus on today is navel orange worm. This is public enemy number 1 for all tree nuts. This probably takes up at least 50% of my research effort in California. So what I want to do today is just give a background on this insect, what modern management looks like in these systems, and then what we're doing to advance that with research and extension work in my lab, as well as with a number of other collaborators. So this insect, considered native to the New World, originally described out in the Caribbean and Central America. You can find it throughout North and South America now. Really wide host range. The name is terrible. It was first described on split citrus in Arizona. That's where it got the name navel orange worm. it's not a direct pest of that crop. They were just infesting split oranges falling on the ground. It really is very specific on, well, not very specific, but it has a strong preference for tree nuts. So you can find it on this wide range of hosts, both agricultural and non-agricultural plants, but boy, it loves walnuts and almonds. And then as we started to develop a pistachio industry in California in the seventies, it moved into that crop as well. So something that's kind of everywhere, a low population, but as we built up these tree nut orchards in California, you kind of created this huge host base for it, and it moved in and just, so early reports from California in the forties, and then on from there. And bear with me, some of these slides I was looking at last night, they're more kind of grower oriented. I think there's a slide showing what the different life stages of a monk are. I assume you all know that. My 5-year-old just learned that. It's preschool, but. So it's a direct pest of tree nuts. They put their eggs directly onto the new crop nuts. The larvae come out, they infest the kernel. That of course impacts yield and quality, but also leads to increased processing time and carryover of moths into the following year, because not all of the nuts are removed from the orchard at harvest because they don't shake out the tree or they pick them off the ground. So kind of multifold issues. Really important is this aflatoxin issue. So infestation by navel orangeworm is associated with the presence of these aspergillus fungi, which produce aflatoxins. These are known human carcinogens, heavily regulated in both domestic and export markets. So for all these reasons, growers have an incredibly low tolerance for infestation by this pest. They're typically aiming for less than 2% infestation. Here's the slide. Guess what? Egg larvae people don't. Okay. As mentioned, they deposit their eggs directly onto the nuts. So just showing kind of the relative scale of that. Split pistachio. Again, all the different life stages and some of the developmental or identifying marks here. What I mostly want to point out with this slide, though, is when they infest the nut, they create this webbing, their frass is in there, so they're not just eating the nut, but they're really kind of the pre-definitive signs of their infestation there. So it's a nocturnal organism. They close just after sunset. Females will start calling them a little bit after midnight. Males will follow pheromone plume, find them, do a little dance, and then they start mating kind of towards sunrise. So studies we've done or our colleagues have done showed that, you know, first night out, they mate, second night out, they start putting down eggs. And so each female will put down around 180 to 225 eggs per female, typically over about a 1-week period. That really starts off high and just slowly climbs over time. So you can imagine kind of the exponential growth of these populations, especially relative to the vast amount of host material available. So here's our UCIPM kind of standard phenology figure. So they overwinter as larvae or pupae in those remnant nuts. Sometimes we'll refer to them as mummy nuts. They can shrivel up, I guess, like a mummy. You get your first flight of moths coming out of those nuts, and then egg deposition of larvae adults again and again, and you get about 4 generations typically per year in California. The ability of this insect to infest tree nuts is mediated by the integrity of the hull. So tree nut, you've got the kernel, surrounded by a shell, surrounded by a hull. And walnuts, for instance, codling moth, different Lepidoptera pest, it can lay its eggs and the larvae can bore through that hole. With navel orangeworm, they can't. They need some sort of outlet opening, whether it's damage from a pathogen or primarily, though, it's really the splitting of these nuts, and that's just part of their natural phenology. So we do know that they have different developmental rates on the new crop nuts versus these remnant old nuts. As you can imagine, graded old nuts, less of a higher quality or lower, it's more of a low quality host material. And we'll come back to this later. This is a really important component of how this insect operates and what's driving some of the research we're doing. We know in almonds and walnuts, it's fairly predictable when this whole breakdown begins. And pistachio, far less is understood about that. So there's some research we've been collaborating on to better understand that. Here's those flights. Typically, you have this protracted emergence in the spring from those remnant nuts. They kind of went into the winter and all these different life stages come out. All they have at that time are these remnant nuts. And so the second flight is always really a big bottleneck for them. There's not a lot of them. They're lower quality, but by the time they get in to that third flight, you know, their populations are booming because now these nuts are available on large scale. So Again, we'll come back to this, it's just a really important part of their population dynamics that, that we're trying to better understand and use to give growers better, predictability, for managing this pest. So these are the key management, practices that are employed right now. So winter sanitation, crop sanitation, I mentioned there's a lot of these remnant mummy nuts that are left behind in the orchard. Really, in pistachio, it's probably the most difficult. Growers need to go in, shake those nuts out of their tree, gather them up, and then mow them or disc them under the soil. Big old walnuts, that's easy enough to do. Almonds are kind of in between. Pistachios, some growers can get them on the ground, get them all in the row middles, go in there and mow them, but it's like, some of them describe it like mowing marbles or something. It's just they're bouncing around, or it's really hard to destroy it. It can be a really big challenge for a lot of growers, and it's important to remove these, not just to get the navel orange worm out of your orchard, but if you leave them in there, it's a reproductive substrate for those that are emerging in the spring. So you could have really low infestation of your remnant nuts, but if you just leave them out there, they could become a source for a population of navel orange worm in your orchard in the spring. This is just some early data. Basically relating, the more of these remnant nuts you have, the higher navel orange worm infestation was observed. So we really, the start of controlling these remnant nuts, it's first off just to get the nuts out of the field as soon as you can. So once a crop is ready to be harvested, we really emphasize that growers just try to just get that thing out of the field. The longer the crop hangs, the more infestation we see, and I'll show some figures about that later. And we can't make the trees grow any faster, of course, but it's really a logistical, managerial type of exercise to get those things out. And then everything I just mentioned, after the harvest, get them all on the ground, get them all in the middle, get them all chopped up or whatever. It's 2025 and we've had some mechanization of this process to get them out of the tree. Like if you go and shake a tree and the nut stays up there, people will go back and shake it again. Typically timing for like after a rain event or something where there's a little more moisture in the nut, gives a little more weight to shake off the tree, but a lot of cases they have to send in crews by hand with poles to knock the nuts off. So as you can imagine, a high labor cost. So get those things down. And the research that's been done so far, a nut hanging up in the tree is probably the best place for one of these navel orange worms to reside. If they go down on the ground, we see more degradation, there's more mortality. If they're on the ground in ground covers, there's kind of even more mortality that's experienced. But even with all that said, we really emphasize growers to go in there, chop these things up, get rid of them. So I have run into growers who said, well, I got my almonds out of the tree, they're down on the ground, probably gonna die over the winter, it's fine. It's like, no, you need to get those out. So monitoring, there's a whole range of stuff that people can do to monitor populations. We have such a low tolerance for damage. We haven't really been able to create economic thresholds. It's been really difficult to relate any of the trap catch to infestation. There's just so many different things going on there that mediate that. Each method provides a little bit different perspective, and we always are encouraging growers just monitor as much as you can in as many different ways as you can. One of the first traps that was developed back in the 70s was this egg trap. It's like a little vial with some ridges on it. Since that really likes texture to oviposit, then they'll put an almond or pistachio meal in there. So this will attract mated females who will then lay their eggs on the traps. Grower sets this up, look at it every week or every few days, count the eggs, scrub them off, and keep looking. That allows you to set a biofix for our navel orange worm phenology models. So when we see increasing egg deposition in the spring, that serves as biofix to then run the degree A models. Growers also use a combination of wing traps and delta traps, baited with either pheromone lures, attracts males, oviposition baits. It's still kind of like what you put in one of those egg traps, so that's bringing in mated females. And then more recently, we've been doing a lot of work with pheromones paired with phenylpropionate, because this allows us to monitor navel orange worm in the presence of mating disruption. So this is more of an important feature of managing this insect. But of course, when you put mating disruption in the orchard, you're going to lose your ability to track the insect with these pheromone lures. So the egg traps and the ovibait lures are great, as I mentioned in the beginning of the season, for setting biofix. But as that new crop comes online, it starts competing with those cues that are coming out of that ovovate, and so you'll still get catch, but it's significantly diminished over the course of the season, although it is still catching something and it does still work under mating disruption, but you know, in contrast, you get these pheromone traps that in the absence of mating disruption, that's, you know, you're getting these really big flight curves later in the season because of the growth of the population. So mating disruption is mentioned, as I'm sure you're all familiar, since they use a pheromone to locate each other to mate. If you can put a synthetic version of that out there, you can interfere with that process. So currently we've got a few different types available. There's 3 different products that use an aerosol emitter like this. One which is using this polymeric emitter, kind of impregnated with the pheromone, and then more recently companies have been marketing this flowable version of the pheromone. It's really the newest thing. The testing that's been done to date, courtesy of David Haviland, a entomology farm advisor out in Kern County near Bakersfield, did a really nice study and showed basically that these 3 aerosol products and polymeric emitter, they all basically work. They're very effective. You know, they can reduce damage by about 50%. They have different costs. They have different ways they set them up. You know, whatever the grower wants. There's different bells and whistles associated with each one. But you know, these are all good products. And the flowable data on that is still kind of out there. We haven't really had it tested thoroughly, but you know, there's still challenges with this. As great as mating disruption has been, and we've had it for about 10 years. You know, you still have this challenge with mated females moving into the blocks under mating disruption. You have the need to, you know, implement this over a large area, which can be a challenge depending on where you are in the state. And then, you know, it does, you know, you are losing the ability to track populations with pheromone traps, and that's kind of been the impetus for us to work on this film for pioneering, as well as kind of pushing forward on some of these ovovate lures, trying to create a synthetic version of those plant volatiles. So insecticides, still a huge important component of management of this insect. The timing is really targeting these changes in whole integrity, less so than the actual population. So when the crop becomes vulnerable, that's when these pesticides go on. And of course, as with any crop, you know, we're always pushing calibration and proper weather conditions for applications. The products that are primarily used today, methoxyfenazide, chlorantranoloprol, that's really the combination that's put in a lot. People still do rely on pyrethroids as well, and spin that around to a lesser extent. So this is an analysis of in California, we have mandatory pesticide use reporting. So we have this database of every year, every block, what was put on, when was it put on, what was the rate. It's a really interesting thing to query. And what I put here, these are just pyrethroids being used in pistachios and almonds, which are used for stink bug and plant bug control, but also for navel orangeworm, as I mentioned. So you can kind of see these peaks here in the spring. That's largely plant bug, and then you get these peaks here around whole split and almonds. Again, you get some spring applications here in pistachios, and then their whole split occurs a little bit later, so you can see that rise and fall. This is a summary of some survey data, and I'll talk more about the survey in a bit. Just showing the number of sprays people reported putting on for navel orangeworm in almonds, pistachios, and walnuts. And then this is drawn from that same database, just showing, you know, as we've had a reduction in organophosphate use, we're seeing the rise of some of these other active ingredients. So again, pyrethroids, methoxyfenazide, really key components to managing insects generally, and navel orangeworm specifically. Timely harvest, as I mentioned earlier, you know, this is a nice data set again for David Haviland out in Kern County. The longer these nuts sit on the tree, the higher percentage damage we observe. It makes sense. They're out there ready to go. You got moths just circulating crazy. So really just emphasizing this with growers to get these nuts out of the trees. And as I mentioned, it's really largely a logistical managerial type of exercise. So, okay. So that's kind of your crash course in navel orangeworm management, just to set the scene a little bit. So going back to this phenology chart, we really emphasize, you know, as much as crop sanitation is considered the foundation of navel orangeworm management, if you only do one thing, that's the one thing I would really push. And in some years where we've had really low prices, growers have been facing that sort of situation. Like I can literally only afford to do one thing. Should I spray half my field with pesticides, should I do this, should I do that, it's a lot of bad choices sometimes, but really the way we talk about this in our extension work is that you really just gotta hit them with everything you've got, so, you know, what I'm doing here is just overlaying these different activities where they are interfering with insect, you know, and it's seasonal phenology, so they're all stuck there on the ground in the winter. Kill as many of them as you can with sanitation. The ones that survive, you know, hit them with mating disruption, make it really difficult for them to find each other and mate. Those that do, hit them with insecticides. Those that make it through that, get that crop out. So just boom, 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 just keep them on the front. It's really what we say. So, so I think we have a nice program for this insect. You know, it's a product of many people, many years of work. But there are still many challenges, and this is really where my research and extension program comes in. So we conducted a survey a few years ago of tree nut growers as well as pest control advisors. Those are the people making like consultants basically on pest management in these crops. What are you doing for native orange worm? What are you not doing for native orange worm? If you're not doing something, what are the main barriers? So kind of a needs assessment. And there's all sorts of interesting things we found there that have, you know, kind of relating to the demographics of the farmers and scale of production and stuff. But some of the key takeaways here. So here's these key management practices: crop sanitation, pesticide sprays. Basically, everyone's doing that. They may not be doing it effectively, they could, but they're doing that. But in contrast, you know, mating disruption, kind of a newer technology, we're not seeing as much adoption of that. With monitoring, you know, evaluating the remnant nuts, we were going out in the orchards, cracking these remnant nuts, trying to get an understanding of, you know, it's not just, you know, do I have 100 or 200 remnant nuts per acre, but what is the actual infestation rate of those nuts? That's of course gonna inform, you know, the potential negative impacts those nuts could have if you don't remove them. Many are running phenology models, many are using pheromone traps, egg traps and ovibate traps. These are kind of starting to see well, this one's falling and this one's rising. A lot of growers report those egg traps. Kind of go count these little eggs, gotta brush them off with a brush. It's just kind of a pain in the neck for a lot of people. Where if these ovibate traps, it's a sticky card, and they just go count these big adult moths. And in some of our, you know, initial evaluations of them, about 95% of the navel orange worm that show up in there are mated or are females, and about 99% are mated. So it's fairly reliable. And one of my colleagues at USDA has been relating egg trap catch to ovibate traps, and so far it's basically showed they kind of track each other. So we may be getting to a situation where this sort of trap setup can replace these egg traps in the future, and I'll show why that might be important here in a minute. So we had that survey, and that was across all tree nuts, and then this past spring, the Almond Board of California, they held a roundtable to discuss, again, like research needs and priorities. And this is not exclusive to almonds. Many almond growers are also pistachio growers, and many of the needs that were identified are relevant to walnut and pistachio growers as well. And so it was like an all-day affair. Everybody's doing all that classic stuff, voting and writing on whiteboards and all this stuff. A lot of broad representation across the industry. But the top priorities that came out of this was a need for regional monitoring, need for better risk prediction, like movement of the moth into the orchard, really kind of encapsulated within the area-wide management program. So that's really something that we're pursuing and that I want to talk about more here today. And this makes sense, you know. Cardinal sent put a video. Oh, okay. So this is not how we run flight mills, right? We just have one moth, not two, but the grad students thought it would be cool. So my colleague Chuck Burks, USDA ARS, he's out there in Parlier, right across the street from the Kearney Ag Center. We collaborate quite a bit. He did the initial flight mill work with navel orange worm, and you know, demonstrated that it's a fairly robust flight. So we know it has a strong flight capacity, and we know it moves between orchards. So some work that was conducted about 10 years ago. Colleague of mine, Steve Bays at UC Berkeley, used these fatty acid markers. So you could basically take an adult moth, look at the ratio of these different fatty acids found in the adult moth, and link that back to the larval host that it's feeding on. They were able to differentiate almonds from walnuts in terms of where the moth originated, and then showed that, you know, indeed, here's walnut orchards where they're finding some fraction of moths from almonds and vice versa. Not a huge surprise, but the first time that this has been, sort of quantified and there was a tool developed. Unfortunately, and we'll talk more about this later, the signature for almonds and pistachios is really similar, and so there's more work that we're pursuing now to better refine this tool. Study that I became involved with early on, again with Chuck Burks at the USDA, using those fatty acid markers to look at movement of moths into walnut orchards. And so what we did initially was to select walnut orchards to monitor that were in very walnut-dominated landscapes. So all the red here, these are walnut orchards, all the black is almond. And then in contrast, in almond-dominated landscapes, here's the walnut orchards, you go monitor therein and see, what is the timing of moths from outside of these walnut orchards? What, percentage of the population they make up. Given the staggered phenology of just 3 tree nuts, you have almonds being harvested first, then pistachios, then walnuts. And so the thinking was, at the end of these harvest events for these other 2 major crops, you have a bunch of navel orangeworm kind of roaming around out there, and they all kind of vector themselves down into walnuts. And so, again, we can't separate almonds and pistachios. It's an admittedly limited tactic or strategy here. But what we can see here, so this is showing the percentage of navel orangeworm that were with a walnut signature. So if you're in a walnut dominated landscape, you're seeing quite a pretty consistent amount. But if you're in this almond dominated landscape, you're seeing changes here over time. And this is kind of coinciding with harvest events in almond, and then potentially, more of the moths, being removed from the system here. So it's not a perfect picture, but just sort of wading into some of this movement work that we're interested in, further elaborating on in my lab. So there's various manuscripts of preparation around this, but the point is, they move. Even early work, so back in the 70s, very kind of straightforward study, but comparing, navel orangeworm populations in an orchard that was adjacent to a very well sanitized almond orchard versus partially sanitized, and showing that there were differences there, and they can't trace it directly there, but trying to get at that point. I guess the idea that movement of this moth into adjacent orchards has been something that researchers have identified for years. This is a more recent study where another USDA scientist called Siegel, they basically had a large data set of crop damage and navel orangeworm populations across a series of orchards. And what they're showing here is that the proximity of an almond orchard to a pistachio orchard seemed to be one of the stronger drivers of infestation. So I mentioned earlier that pistachio orchards are really difficult to sanitize because the nuts are so small and durable. So this is kind of their argument, saying, look, generally if there's a pistachio orchard nearby, even if the grower's trying, they're still going to miss some. And, we tended to see a relationship here with infestation. So all that said, the timing, the extent, and catalyzing forces of this movement are really, so this has become a major theme of research in my lab. And, to address that, we're having to develop new tools that may even ask the questions that we want to ask. So I mentioned I was going to come back to this thing about tree phenology and host availability. So here's those flight curves again. So in the spring, all you got are these remnant nuts, poor quality hosts, low abundance hosts. But then you have around July 1st, the new crop almonds, they experience whole split. It's this, that out of all the nuts, we have like 1 million+ acres of just almonds. So it's this massive, introduction of viable host material. And so you get populations just sort of exploding off of that. But then you have harvest, and that's this mass removal of host material from the landscape. And I mentioned staggered phenology, so whole integrity begins to break down pistachios in the middle of August. So another mass introduction of resources, but then harvest, that's removal. And then as you can imagine, the final one is walnut. Now, obviously, it's not this black and white, just like one on, one off, one on, one off, but they are definitely stacked. I think this is an important dynamic to better understand. So, when you look at this map I was showing earlier, where all these tree nuts are throughout the state, 2 million+ acres, they're not all necessarily readily available to this moth at the same time. So this is just another iteration of one of these crop maps showing specifically where almonds are, and pistachios, and walnuts, and you can see there's different aggregations and different combinations of the three nuts. And so, you know, part of my background is in landscape ecology, insect movement, area-wide IPM, and so, you know, we really think a lot about this in the lab, like, how do the moths move in these different landscapes? You know, what is your vulnerability if you're an almond grower here versus, you know, all these other situations, so. Much less, what about harvest time? What about mating disruption? What about pesticide sprays and crop sanitation? And I know some of this stuff is hard for us to capture, but it's stuff that we think is important and could be driving the populations and ultimately determining how much risk a grower has for these insects moving in. On top of that, I mentioned with mating disruption, you know, there's serious challenges in getting more people to adopt this, and I think this is in part due to the scale of operation we have. So in West Fresno County, very large blocks. These are all 640, 160 acre blocks here, right? So very large aggregation of blocks managed by a small number of people. Versus in Stanislaus County, this is closer to Modesto, a little bit north of Fresno, you have these aggregations of all these tiny little growers, 40 acres, 60 acres, 80 acres, a lot of historical family farming operations there. And when we looked at our survey data, we did see that adoption of mating disruption tended to be higher with these larger operations. As you can imagine, when we talk to these smaller guys and say, look, there's 40 acres, spend all this money for mating disruption, all these guys around me aren't doing anything, moths are coming in, you know, it's $125 an acre for me to set this product up. It just doesn't pencil out for them. So there's a need, we think, to figure out how to facilitate the formation of these aggregated groups of growers that would adopt mating disruption. So all that said, right, there's this industry meeting, we want more area-wide, there's kind of this movement argument for why area-wide management makes sense for this insect, and the facilitation of not just regional monitoring, but regional coordination and scaling of best management practices. And so we have recently launched a new project. It's a collaboration between Riverside, UCIPM statewide program, Washington State University, and USDA ARS, along with our industry partners, so California Almond Board, Walnut Board, California Pistachio Research Board, and American Pistachio Growers, kind of a parallel organization. And that's, you know, myself, Chuck Burks, I've mentioned a number of times, Jolinda Rajal with UCIPM, and Dave Crowder of the Washington State, of course, these two partners that I already mentioned. So what we're proposing and what we're actually putting together already is this online tool that's going to provide regional monitoring, phenology estimation, both for the insect and the crop, as well as some ability to coordinate adoption of best practice. So this decision aid system, this thing has been around. How many people have heard of this? Okay, all right, so I'm bringing something kind of new. You know, this was started out of a kind of a West Coast collaborative project, Washington, Oregon. All the entomologists that were working in orchard crops at the time. And the idea was to create this online tool. And, you know, they developed it for codling moth in apple, the major crop in the Pacific Northwest, kind of a minor crop in California, but codling moths, pests, and walnuts, there was impetus to do this. And really, in my experience, it seems like the California group, most of those folks kind of retired and the system was built and it just stayed up in Washington and just kind of became this Washington thing. But with Dave taking over and kind of he and I having shared interest around this, we saw this as a real opportunity to expand, kind of leverage their experience with developing this type of system for navel orangeworm in California. So they did the whole thing with codling moth, but now there's more than 50 pests and crop combinations across different regions of the US. I mean, it's still primarily Pacific Northwest thing, but they're doing monitoring in Idaho and versions of Wisconsin and continue to build this out, of course, with partners in all those areas. So you can check out their website, kind of look around there. What this thing does is, well, there's numerous things. So, and just really depends on what the industry wants to set up and what the researchers are able to drive forward. So regional phenology monitoring, degree day and life stage, phenology models, putting it into kind of a visual format, risk assessment, a number of different things. They basically have a team of programmers that as we talk through the issues with them, they can come up with basically whatever we want to display. So the pistachio and almond industry put some money forward last year to get a pilot of this setup. And so just a very basic readout showing degree day accumulation, putting your biofix here. It's kind of a narrative readout of what conditions are and what the management recommendations are. This is not functional at this point, just kind of a pilot to start beta testing, talk with growers in the industry about. But the way we're approaching this, it's kind of twofold, and this will kind of be the rest of the talk here. In the short term, we want to take the existing tools for naval orange management, make them easier to use, and make it easier for growers to facilitate the adoption and scaling of these practices. And then in the long term, we want to develop new tools in this platform, or you kind of centered around this platform. So I'll just step through both of these here. So currently, we have a phenology model for naval orangeworm. It's hosted on the UCIPM website. It's kind of like something that they made in the late 90s and have not updated since then. And people use it, but the feedback we've gotten is that it's a little bit, it's not very user friendly. So you got to put in your biofix, you got to select your weather station, you got to use drop down menus with dates and everything. And it puts out this table of accumulated degree days. And this is based off the CIMMYS network. It's for irrigation management. You have these weather stations that were set up in the 80s, and they're all throughout California. But a number of growers have said, these weather stations aren't necessarily representative of my conditions in my part of the county. The way they're set up doesn't really reflect how an orchard would be experiencing weather and climate. And then on top of that, the growers got to go through that table and find, there's 1,050 degree days. Oh, okay, there's that. And, again, some guys love this, but others, a lot of complaint about it. So there's a need to like, it's not that the model's wrong. It's just we could have better weather data, more user-friendly. And in the same way, we have crop phenology models hosted primarily through the UC Davis Fruit and Nut Research and Info Center. Pulse split calculator, pistachio bloom forecasting, a number of other things. And what feedback we've heard from growers is like, we're trying to relate the tree phenology to the insect phenology. We have to go over here for this model, we have to go over here for this model and put it together. And, again, we just want this to be an easier user experience. And so what we're in the process of doing is moving all those models over to this new platform. There's a map interface where growers can save specific locations. We're using higher resolution weather data. We're still giving the table as an output because some people do seem to like it, but also including more of a visual representation of, here's your cumulative degree days, here's flights would occur. This is just kind of a first draft, but I hope you get the idea. More to it to use. What we also want to do is integrate these models so that you could actually forecast over the landscape where you might have greater or lesser vulnerability to this insect. So crop vulnerability, low versus high, native orangeworm population, high versus low, simplistic thing, but what I'm trying to emphasize is that there's going to be certain regions where if you've got a lot of hole degradation, a lot of native orangeworm, that's a problem. If you've got pretty good hole integrity and not very many native orangeworm, maybe not as big of a problem. And so growers can prioritize where they focus their management and monitoring efforts based on the readout from these models. So the final bit is linking management recommendations to these model outputs. So the UCIPM website, that's where we center all of our monitoring and management recommendations for these different insect pests, not just of tree nuts, but of all crops in California. And it's great, it's regularly refreshed and updated, but what we're coming up with, it's not new management recommendations, it's just pulling from that website over to this new platform and giving a little bit more of a narrative readout. So this is for codling moth, we haven't fully developed the native orangeworm yet, but they put in some phenology info here. Emergence is expected at 175 degree days. That's going to be 12 days. Historically, the first moth is caught between these dates, and studies show it's just a little bit more interpretive, representation of what's going on. So that's also paired here for codling moth. They have the different active ingredients that can be used, what their impact on natural enemies is. Like they've got kind of the most advanced version of this for codling moth. We're hoping to bring some elements of that to enable large worm. So those are some of the short-term things we want to do. But where my interest really lies is in this longer term development of new tools to help further manage risk for this pest. So here's another crop map. One of the things we're trying to work on now is developing a regional monitoring program based in the use of automated smart traps. So the idea being you could run a network of these traps, which UC Cooperative Extension historically has done for a number of different pests in different regions. I did it as a postdoc running an area-wide program in vineyards. It's a lot of sticky traps, a lot of driving around, writing stuff down, putting it into a table, putting it in a newsletter. It would be great if we could do this in a little less labor-intensive way, and then use that trap catch data, whether it's daily or weekly or however you want to iterate it, to give an update on where population's high. So currently we are evaluating a series of different smart traps that are out on the market. This is just a sampling of some of them: FarmSense, CropView, TrapView, Scout Labs, and we're putting both these Avivate lures, and then we're also testing them with the pheromone and phenol lure, and comparing them to our traditional Delta and wing trap lures. So we're comparing these both for accuracy of insect identification. So that's like an image from one of their images or pictures that count on moths. But then also seeing if the phenology curves these automated traps generate are similar to what we get from a traditional trap. So we're testing them across a series of 9 orchards, 3 pistachio, 3 almond, 3 walnut. And what we've seen so far, this is just a quick summary of the year 1 data. Fairly accurate, the FarmSense one a little less so. The reliability varies, so they're connectivity and giving daily updates and access to cell phone networks and stuff, and cost varies. So it's not just how accurate are they, but also just how user friendly these things are, how affordable they are. If we're wanting to scale this, we can't have a trap that costs $650 per year. It's just not going to work. And then, so now we're kind of wrapping up our second year of study with looking at the accuracy, and then we're also going to start working on other field trials to look at trap spacing and how you best interpolate data between these gridded trap points. So all of which is kind of working towards how do we, if we're going to do regional monitoring of these traps, how far apart do we space them, which trap do we use, etc. Something else we want to work on eventually is using those automated traps to potentially set a biofix automatically, which could feed into this phenology model. So right now, growers need to go look at a trap, whether it's the egg trap or an Avivate trap with a sticky liner. And when you see 2 weeks of increased egg deposition or capture of gravid females, you can go back 2 weeks and set a biofix. So it seems like we could do that with the automated trap catch. And similarly, most of these crop phenology models are based on their bloom date, which I think in most cases we should be able to remote sense with like daily satellite imagery, so that we can say, okay, the almonds in this region are ahead of over here. And, again, right now growers can go and they'll be able to enter their own biofix states, but for regional monitoring, we really would like to find a way to capture that across not just an individual field, but a series of fields in a region. So yeah, that's the point, iterating there. I think I got a little bit ahead of myself here, where this is what I was just talking about, bloom and the automated catch. So the regional coordination piece, the way we're thinking about it now is that we could use parcel maps that have ownership and even crop type tied to it, and create a process by which growers could go in. They'll each have their own account, and I should mention this decision aid system. This is totally free, but individual growers set up their own accounts and then go in there and put their little weather stations at their blocks. And what they would then also be able to do is claim their parcels. Oops. and indicate their willingness to participate in one of these neighborhood groups or regional coordinated groups. So just an example, people kind of raising their hands saying, okay, I'm here, you're here. We haven't defined, what counts as a neighborhood group. So I don't know, say 1,000 acres right now. And if they get enough of a group together, then it unlocks their ability to access various incentives for not just adoption of mating destruction, but also potentially other best management practices. Right now, we've been working with a few manufacturers to set up some sort of a rebate program. So if we can go to a company like Trace and say, look, our platform is going to deliver 5,000 acres of interested growers, could you provide them a discount? So far, we've gotten a positive response there. We'll keep working at that. The NRCS EQIP, Environmental Quality Incentives Program, does have payments for adoption of mating disruption specifically. There are income caps on this program, which make it a little bit difficult for some of our growers to enroll or qualify. There is a version of this, a regional conservation and planning program, I think is what it's called, a partnership program, where you can sort of get around those a little bit. In California, we do have legislation that if enough growers in a region band together and want to create a pest management district, they can actually start taxing themselves and use that money to either offset mating disruption or pay for regional monitoring. We haven't come down on which combination of these we're going to use, but just saying there are various financial incentives that we can use to motivate this. We are considering, if you enroll in one of these programs, you acquire these incentives, you do need to provide some basic details on your block, the sanitation levels. We'd have like a standardized survey for them to figure that out. Pesticide use, they can either report that or we can get it directly from the pesticide use reporting database. And most importantly for us, crop damage. What we're thinking is that this might be an opportunity for us to aggregate a large data set on pest populations, management practices, and then outcomes via crop damage. And then we might actually be able to start doing some additional analysis to understand, was crop damage high in this region just because that region has a lot of moths, or is it because of certain combinations and timing practices that growers were implementing to control it? There's a lot more I think we could do here. Again, we're just kind of rolling this out now. We have a very motivated group of growers in these two regions of the San Joaquin Valley who have expressed interest and willingness to provide this type of information. I think on top of, all the financial incentives I'm talking about here, there is, in California especially, always this looming potential for regulation of key insecticides. And we have a new kind of emphasis on that. I'll talk about it at the end recently. And a number of growers have said, this sort of program, they think there's also some beneficial optics that may come out of that. So if the state's looking around at, where do we need to regulate pesticides first? I think the tree nut industry in California wants to be able to say, look, we've created this new tool. We are organized. We are scaling these things. We are doing the most we can to get everybody to do all the best management practices. We'll see. That's more of a political thing, but it has been mentioned in our extension meeting. So this navel orangeworm movement piece, I keep talking about getting better risk prediction. So current limitations, I mentioned already, fatty acid markers, they only differentiate between almonds and walnuts. And even when you do know the origin of that moth, you don't know if it came from next door or down the road or all the way across the valley. So there's a lot of other questions there and things that we're trying to develop. So in order to answer these questions, we've set out to develop a number of new tools. So kind of a second run at the fatty acids effort, a graduate student of mine, using kind of a broader scanning of all the lipids they're finding in these insects to potentially find something unique to pistachio. We've also looked at compound-specific isotope analysis, as well as mitochondrial DNA, so Vermont Bonsall at the USDA. And then we have artificial markers. So that first one, it's like we just want to be able to capture a moth, squish it, put it in a machine, and it's going to say, what crop will this thing come from? And then these artificial markers, we've got two different categories. One, indirect, where we mark the tree. We've been working with N15 as well as rubidium and had good results, promising results so far. We're going to start moving that into more moving it from proof of concept to actual field applications. And then direct marking of the moth itself. So there's been a lot of work, postdoc is focused on this smart water. It's like a UV reactive dye. Historically, we use like a fluorescent powder to mark these moths externally. With this smart water, it's like, put it into a nebulizer, it creates like a fog that goes over the insect and, does a pretty good job of marking them with minimal impacts on the performance. And then most recently, we were contacted by somebody who wanted to talk about harmonic radar, and they've got some of these tags. They're putting them on mosquitoes now, I guess. They're really getting some nice, advances there. We tried this with some bigger insects years ago. We'll see where that goes. But anyways, just to show there's kind of a broad spectrum of things we've been looking into. The MT, the mitochondrial DNA, this has been interesting with high throughput sequencing. Again, Ramon's the guy leading this. We're able to see trace amounts of DNA from specific host plants in these moths, which we verified, reared them on that host and pulled them off. And then this is some data, again, from Ramon, where we collected navel orangeworm from very walnut-dominated landscapes, pistachio-dominated almond, and, the moths that have more almond, tended to have more almond DNA to live out of these almond, concentrated almond production areas. So, still kind of proof of concept, there's cost issues and things like that, so there's more to come here, but just showing a little sample of some of the stuff we've been doing. And then how are we going to use these tools? There's a number of different things we'd like to do. The rubidium, you can apply that to trees and then see when and how far the moths are out from a given orchard or orchard area. Larval host signature, so how does the composition of the landscape affect the timing of who's coming in there? Of course, still questions about, how far do they fly? There's some flight energetics and moth nutrition work that we're starting to do that, hopefully we would love to be able to not just pick a moth off the trap and say, oh, you came from almond and boy, you flew a lot before you got here versus flew a little. And even then I get it, moths can fly really far in a straight line or do this in a little orchard and burn calories in a different way. But anyways, getting at some of these questions with these tools and then mark release for capture, within a block, how do moths move around in space here within a confined area versus going out. So yeah, that's kind of this big area-wide project and online platform we're launching and a number of different research projects we've been developing around insect movement at the landscape scale. And going back to this figure, we certainly have a nice program in place for navel orangeworm. There's of course challenges with adoption of these current practices, and as I alluded to earlier, there is going to be a continued need to develop non-pesticide alternatives. So the Department of Pesticide Regulation recently convened a giant stakeholder group. It came out with this document, the Sustainable Pest Management Roadmap. And the key thing they're trying to do is reduce the use of the most toxic pesticides by 90% by 2040 or something like that. They haven't identified what those pesticides will be, but the fact is there's a lot of pressure coming in addition to the usual pressure we see in California around pesticide regulation. So there's a lot of new research in addition to what I just presented today that we're involved with, looking at grazing and winter cover crops for crop sanitation, new equipment for that. There's a whole program on sterile insect technique, a lot of the whole integrity models, we're trying to improve those, and of course screening, continuing to screen novel chemistry and compounds for chemical control. So with that, I just want to acknowledge all the funding we received for various aspects of what I presented today from a number of different commodity boards and state and federal agencies. Again, huge honor to present to share a little bit about my work here, and happy to take any questions. You got my contact and website. So thanks so much. Thank you.
Before taking any question, I have a quick announcement. We are hosting a social hour with Houston at 5:30 p.m. at Swamp Pit Brewery. Please feel free to stop by, and we can take 2 questions. Yes, of time. For everyone.
Do you see any potential for corporate sponsorship in order to reduce some of the costs of like mating disruption, adoption, or something like that?
Well, so the incentive, the rebate program, that's something we're negotiating with mating construction companies right now. Is that kind of what you mean,
I
or
was
is
just
this?
thinking of the Cliff sponsorship
Oh,
of
right.
your research institute. Do you like something like that possibly?
Yeah, we haven't pursued that yet, but I don't think something like that is necessarily off the table at this point. It's very early stages for all this, and right now we've been thinking about this platform and how to pay for it over time. The commodity boards have expressed some interest in partnership with UCIP, maybe that they would help support that. Now that doesn't offset mating disruption, right? It's the companies that provide a rebate for that, but yeah, there's a lot of growers. There's only 2-3% certified organic. But a lot of growers are telling us, we sell to Mars or some of these other Danone or somebody like that, and they have their own kind of sustainability programs that they have in place, and this might be something that they could tie into and say, look, if you're involved in this area-wide program, we'll pay you a penny more per pound or half a penny more per pound. So there may be some other additional incentive we can put in there. So,
I've been hearing about the sterile insect technique program for a while. Is it moving forward or is it stopped or what's the status?
yeah, it's definitely moving forward, and we're highly involved in it. There's kind of like two different pieces to it. So they have this large facility in Phoenix, Arizona, where they're mass producing sterile egg larvae, about 1 million or 2 million per day, depending on their resources they get together. USDA APHIS leads that in partnership with California Department of Food and Ag. They've been running what they call pilot programs, so they're releasing moths over a number of blocks and looking at crop damage. What we have been doing on the side is evaluating the quality of those moths, the dispersal of those moths, like how do they compare to a locally reared, non-irradiated moth. We've also been looking at the development of alternative non-nuclear irradiation strategies, so a lot of X-ray work, a lot of electron beam work. And the mass rearing facility, I think the industry is set to kind of transition into operating that in the next 5 or 6 years. That's a kind of a roadmap that the Department of Food and Ag put out recently. So yeah, that work, we don't have a program that, growers can't buy sterile moths and release them on the farm at this point, but it's, as research goes, kind of making incremental, progress as we go. So we'll see. There's some modeling exercises that we're hoping to do soon that given 2 million acres of tree nuts, given how sterile insect technique works, are there certain regions of the state where it would be better to use this versus some other practices? What would be the costs associated with that? So, yeah, that's as succinct of an answer I give you. We can talk more for sure.
So thank you so much for attending and this
That's my one.
Yes, thanks.
I very.
Pick it up.